Today we are going to talk about lambda based cloning vector, right? In the last segment we talk about M13. It is also a type of bacteriophage. Lambda is also another type of bacteriophage, Lambda Bacteriophage. Here we can see it is a typical lambda cloning vector. It has ori and different sites, right? So before talking about lambda based cloning vector, we there were two problems that had to be solved, right? First was the size limit, right? And as we know that lambda DNA molecule can be increased in size by only 5%, representing the addition of only 3 kilobase pair of new DNA. If more than 3 kilobase pair of new DNA, that is more than 52 kilobase pair in total after adding, then it cannot be packaged into the lambda head structure, right? So this limits the size of the DNA that can be inserted into an unmodified lambda vector. Here, as you can see, normal lambda genome, 49 kilobase pair, we assume it can be packaged. But if assume that new DNA is more than 3 kilobase pair, total will be more than obviously 52. It will be too big to package into the lambda head structure, right? And second, we will talk about multiple restriction sites. Multiple restriction sites. And this the lambda genome is too large that it has more than one recognition sequence for virtually every restriction endonuclease. Right? Restriction endonuclease cannot be used to cleave normal lambda genome, that is to allow the insertion of the new DNA, because lambda genome would be cut into several small fragments, and if they were cut into several small fragments, that would be very unlikely to reform, right? That would be very unlikely to reform to form a viable lambda genome on religation. Here we can see there are multiple recognition sites for the eco-R1, and the eco-R1 came, then it cleaves at lambda genome into multiple, but due to which the religation will be very complex. It will be a mixture of molecules which will be very complex to religate or reform to form a viable lambda genome, right? Second, now we will talk about how to tackle this problems, right? So researchers first did this segments of the lambda genome that can be deleted without impairing its viability, right? A large segment in central region of the lambda molecule can be removed without affecting the ability of the phage, right? Without affecting the ability of the phage. Here we can see this region can be removed, right? Removal of this part will not affect that much. Removal of all or part of that non-essential region that I just shown you between the positions will decrease the size of the resulting lambda molecule by up to 15 kilobase pair, right? This means as much as 18 kilobase pair, like 15 plus 3, right? I just told, right? So this means that as much as 18 kilobase pair of the new DNA can now be added before the cutoff point for the packaging is reached. And this non-essential region in fact contains most of the genes for integration and excision. And integration and excision I just showed between the region that we deleted or removed contain the integration and excision of the lambda prophage from the E. coli chromosome. A deleted lambda here, it is not genes, it is genes. Sorry for the mistake, right? Yes. So I told you that deleted lambda genome is therefore non-lysogenic. Therefore non-lysogenic. There are two cycles, lytic and lysogenic cycle. But here the deleted lambda genome will be therefore non-lysogenic due to the deletion of the integration and excision part of the lambda prophage. It will only follow the lytic cycle, right? Lytic infection cycle, which itself is desirable, as we know that for the cloning vector, which means induction is not needed before plaques are formed, right? Here I will show about it. Then we will talk about second, natural selection. Now we can use natural selection to be for the lack of the certain restriction sites. We can use to isolate the modified lambda that lack certain restriction sites. We don't need them, right? We don't need them that lack restriction sites for the endonucleases. Now even a deleted or a lambda genome with the non-essential region removed has multiple recognition sites for most restriction endonucleases. For most restriction endonuclease, right? This is a problem that is often countered when a new vector is being developed, right? If just one or two site needs to be removed, then we can use the in vitro mutagenesis, the process called as a method or process, whatever. In vitro mutagenesis can be used, right? I will write here for the in vitro mutagenesis. So what is this in vitro mutagenesis? In this, for example, suppose an EcoR1 site GAATTC could be changed, right? GAATTC could be changed to GGATTC. As we know that GAATTC is for what? EcoR1. It is a recognition site for EcoR1 GAATTC. But GGATTC is not recognized by that enzyme, precision endonuclease, EcoR1. However, in vitro mutagenesis, it was in its infancy. NEISIS. Let me write that. SIS. So, and I was saying that in vitro mutagenesis, it was in its infancy when the first lambda vectors were under construction, and today it will not be much help at that time, right, for more than few sites in a single molecule. So instead, let me rub this. So instead, natural selection was used to provide the strains of lambda that lack the unwanted restriction sites. Natural selection can be brought into play by using a host on the E. coli strain that produces EcoR1. Most lambda DNA molecule that invade the cell are destroyed by this restriction endonuclease, but a few survive and produce plaques, right? They produce plaques. And where does as we have known that they produce these plaques on X-gal agar, right? X-gal agar. And as we know that X-gal agar, it is made up of 5-bromo, 4-chloro, 3-indonyl, beta D galactosidase, right? Remember this. I will write it X-gal agar here. What does that when this gene codes for beta galactosidase, it breaks down the X-gal, that is the 5-bromo 4-chloro 3-indonyl beta D galactosidase, to produce plaques which are blue in color, or depends on the further, right? So instead the natural selection was used to provide this strains, right? But a few due to which the DNA, that molecule that invade the cell, is destroyed, that lambda DNA molecule destroyed by this restriction endonuclease, right? Now several cycles of infection will eventually result in the lambda molecules that lack all or most of the EcoR1 sites. Thank you.